2018 onwards. And this is the 6th time we are conducting the series of Nobel Prize. 2018, 19, 20, 21, 22, 23. The 6th time.
Can you please keep the mic close?
So 9 lectures are Nobel Prize out of 65. So let me introduce give a short introduction to Tamil Nadu Science Forum. TNS of Tamil Nadu Science Forum was formed in 1980 by few scientists and researchers, and one of them is sitting in the founder member of Tamil Nadu Science Forum. Initially, the TNS was conducting hall meetings dealing with higher sciences. At some point of time, in middle of 80, it got out of the hall and it go to the people. We thought that we can go to the people and we can propagate the science rather than calling them to the halls and introducing the science. That way, there was a long march and science lectures happened from Kanyakumari to Chennai, and it culminated at an event called the Arivoli. So basically, so it created awareness about the literacy, and the Science Forum seriously participated in the literacy campaign. The literacy campaign expanded the Tamil Nadu Science Forum throughout Tamil Nadu, and it became a state level body now. So we have a state level office, state level, district level body, and the individual branch. PSL, popular science lecture, is one among the branch of Tamil Nadu Science Forum, working under TNS of South Chennai. South Chennai is a district. So that way it is organized in the state level. We got 6 verticals. So one is the science propagation, which we are doing. Other one is the women in science, the summum, what we call it as summum. We encourage and more women scientists and more people, more women involved in science. That's the problem called summum. And the third one is science publication. So we are publishing a couple of magazines, one in Tamil, other one is in English. In Tamil, it is a Tolib, that's meant for school children, and English is the other one that is for college students. Apart from that, the publishing, we also publish a lot of popular science books in Tamil. So it has a separate science publication division in Tamil Nadu. And the fourth one is we are doing research on environment and development. So that's a separate group working on it. And fifth one is the Youth for Science. So there are 6 divisions we are operating. Each district is functioning with the 6 different participating in all 6 activities. So popularization of science is one among the activities, and PS is part of it. That way, TNS is organized. But I want to make use of this opportunity to invite you all to participate in TNS, because we have been conducting the lecture for several times, and at some point of time, somebody should replace us. We want more people, more youngsters to come and occupy this position, and they have to take charge of carrying forward this. We, a member of Tamil Nadu Science Forum, participate in PSL. So we have also provided the link for registration. We also provided the link for the feedback. So at the end of the meeting, I request you to provide your feedback. Apart from that, we have a Google group meant for PSL. We also have several WhatsApp groups. We also have the Telegram group. All these groups, we have links. All of you should join any of those groups so that you will get all the information about the activities of the conference. With this, let me move to this program. So it is a fascinating research on mRNA vaccine, which will be going to be done by Dr. Elizabeth Hanna. So I don't want to talk much about it, and I invite Dr. Aravindan from Genetics Department of the Madras University to introduce the speaker. After that, speaker will continue with the lecture, and we will have a question and answer session. After that, we will be providing the question. Please, Aravindan.
One and all, it gives me immense pleasure to introduce an author speaker to this audience, Dr. Elizabeth Hanna. So I don't know, like most of them I can't be knowing, but she was my senior when I was doing my PhD. Both of us are from the same institute, National Institute for Tuberculosis Research, NART. It is a prestigious ICMR institute which has been working, which has been functioning in Chennai for quite a long time. And we also have other connections. She is an alumni of the present department where I am currently working, Department of Genetics. So I know Dr. Hanna for more than 20 years now, so I really don't have to have a CV, like to introduce herself. So she has been there at NART for more than 20 years now. She joined there as a SRF, okay, and by pure hard work and productive work, she then became a research associate, and then she became a scientist, and now she is one of the topmost scientists at NART. She is the head of the division, HIV division, and she has several awards. She has got the Best Woman Researcher Award, and this is something that I got to know very recently. And she has also got RO and grant. RO grants, like it is given by NIH, and it is very, very highly competitive and very prestigious grant. So she has got RO and grant. And right from the UG to PG days, she has been a topper in her class. She has got a Best Student Awards. Okay, she has more than 150 publications. She has got more than 6 to 7 funded projects to her credit. And I had the opportunity to be an excellent examiner to one of the PhD scholars who had done a tremendous piece of work. And then there were quite a few of our students who actually got involved and then did research on reverse vaccinology. Okay, so she might be talking a few things about that one. So reverse vaccinology is a cost afternoon thing where you bring in two different disciplines, namely bioinformatics and vaccinology. So I can go on and on, like the CV goes for almost 20 pages, but I really do not want to, waste all your time and enthusiasm. So with this highly abridged introduction, I would like to now request upon our speaker to take the highest and deliver the pitch. Thank you.
Yes.
So at the outset, I would like to thank some science students, all the office bearers. Particularly, I would like to express my thanks to Professor Krishnaswamy, Mr. Vijay, for having invited me here. And I feel it's a real honor to be present to give this lecture today. And I also would like to thank especially Dr. Abhinand for his kind introduction. So going to the talk of today, mRNA vaccine became the talk of the town about 2 years back when the coronavirus was at its peak, creating havoc for the world. And today again, it is the talk of the town because the Nobel Prize for Physiology or Medicine in the year 2023 has been given to two distinguished scientists who have made key contributions to the development of the mRNA vaccine. That's what we're going to see about today. Coming to vaccines, that vaccination is one of the most important things to mankind. Vaccines have revolutionized human health and society during the last 2 centuries. Some life-threatening diseases like polio and smallpox have been eradicated due to vaccines, and so many millions of lives have been saved. So WHO gives an estimate saying that 2 to 3 million lives are saved every year due to vaccines from infectious diseases like measles, influenza, tetanus, and pertussis. So that's the gift that vaccines have brought to mankind. So what are these vaccines and how do they work? Vaccines are nothing but whole pathogens which are dispersed in organisms or their components which when administered into the body of an individual invoke an immune response that is capable of recognizing as well as remembering the pathogen. So subsequently, when the person encounters the real virus or bacteria, the prime immune system immediately kicks into action and a robust immunity eliminates the pathogen, thereby preventing disease. Next slide. So, vaccine research has come come through a number of milestones, and one of them is the development of different kinds of vaccines. So one type of vaccine is the weakened or attenuated vaccine, where the pathogen is weakened, so that it multiplies very slowly within the body, so that it is not able to cause disease. However, it is still able to generate immune responses which are protective in nature. The examples of this type of vaccine are the measles, mumps, rubella vaccine, rotavirus, chickenpox, and an intranasal form of the influenza vaccine. Then we have the next type of vaccines which are the inactivated or killed vaccines. Here, the whole organism is given as a vaccine after it is being killed using chemical or physical methods. And examples of this kind of vaccine are hepatitis A, polio, rabies, and an injectable form of influenza. So the advantage of this type of vaccine is that it can be safely given to individuals with a compromised immune system, like cancer patients or individuals with HIV/AIDS, unlike the weakened viruses, which are not safe for administration to individuals with immune system. Then we have another type of vaccines, which are the pulmonary vaccines. And these are nothing but components of the pathogen. It could either be a protein or polysaccharide coated pathogen, which is used as a supplementary vaccine, or like for example in the hepatitis B shingles or HPV vaccine, or it could be the toxin of the vaccine, which is first converted into a harmless non-toxic form or a toxoid, and used as a vaccine as in the case of the EPV vaccine. And then there are also vaccines which use the genetic components of the pathogen, like the DNA and the RNA. And now we have the new kid on the block, which is the mRNA vaccine, about which we will be speaking. So before we look at mRNA vaccine, just to briefly look at what mRNA is, we know that mRNA is an essential component of the central dogma. It is an intermediate molecule which translates the genetic information which is encoded in the DNA to form functional proteins which are important for various life processes. So since the mRNA has the blueprint of a protein, it means that if you use mRNA to produce a protein, any specific protein of a pathogen to be used as a drug or a vaccine. So this is the principle of the design of an mRNA vaccine. So when we talk about an mRNA, it's not just a piece of genetic material. It has got a distinct unique structure. And the most important component is the coding sequence, which codes for a particular protein. But it is flanked on either sides by regulatory sequences called the right time and the time untranslated regions. These are important regions containing regulatory elements which are responsible for expression as well as the stability of the protein. And also, we have additional components such as the cap, which protects the mRNA at the 5' end, and also guides it to the ribosome to initiate protein synthesis. And finally, it is capped by a series of adenine residues called the poly-A, which again has got a protective nature, protects the mRNA from being created from RNases, and also stabilizes the RNA molecule, so that when it enters the host cell, it is able to express itself in the form of protein and initiating immune response. So the mRNA vaccines didn't come just like that during the coronavirus pandemic. They have a long history. So in the next few minutes, I will just go through the history of development of the mRNA vaccine, just highlighting some of the key contributors to the development of the vaccine. So here in the picture, you can see a person. His name was Robert Malone, and he was a graduate student at the Salk Institute for Biological Studies at Washington, California. And during the year 1987, he was pursuing his PhD there, and he did a very landmark experiment which is worth seeing. And in this experiment, he took strands of mRNA, mixed it with tiny fat droplets, and formed a kind of molecular stew. And then what he did was he took human cells and bathed them in this molecular stew. He found that very interestingly, the cells absorbed all the RNA and started producing the protein for which the mRNA coded. So Malone immediately realized that this was a very important discovery with far-reaching potential in medicine. So what he did next, right? He immediately took down his lab notebook, jotted down the notes of the experiment. He signed on that page and dated it as 11th January 1988. Not only that, he requested one of his lab mates to also sign and date the page. So such was his enthusiasm and belief that this was a very remarkable discovery that he had made. Because he wrote down saying if cells could create proteins from mRNA delivered to them, it might be possible to treat the RNA as a drug. So he had the vision at that point in time. Malone's experiment itself did not come like that. It had actually drew from the research of several researchers who had worked before. So as far back as 1978, scientists had used fatty membrane-like structures, which are known as liposomes, for coating as well as transporting mRNA into mouse and human cells to produce proteins. And this work itself had depended on the work of so many researchers who were working on mRNA as well as liposomes, both of which were discovered in the early 1960s. So this is a long history behind Malone's discovery, but we can still say that Malone's landmark experiment formed a stepping stone for the development of the mRNA vaccine. Even this way, success did not come just like that. Malone had difficulties. In 1989, he had a problem with the supervisor, and the person shown there, Professor Inder Verma. So the cliff was quite strong. So Malone had quit his studies, and he actually never went on to earn a PhD degree. But his passion for research was still alive. So he went and joined another researcher by name Fellner. And Fellner had at this time established a small startup company by the name Vital. So he went there and started working with Fellner on mRNA vaccines again. So Fellner also had collaborators working with him at the University of Wisconsin. So together they started working on lipid mRNA complexes, and here showed that they could spur protein production in mice. So again, things started getting messy for Malone. Both Vital working with Wisconsin, where Malone currently is working, as well as Salk in University, where he worked previously. Both of them started filing for patent for this discovery in March 1989. So both parties did not know how to solve this, but due to some unexplained reason, Salk suddenly decided after a year to withdraw its claim, to abandon its claim. So obviously the patent rights went to Vital. But something else also happened in between. Professor Inder Verma, who was still at Salk, joined Vital's advisory board. So Malone guesses that something has transpired. So he was again driven to frustration because he was only mentioned as one among the several inventors, and he did not gain Vital licensing deals, which happened subsequently. So he left Vital. But he went on and did a good thing. He joined medical school, completed his medical degree, did a year of clinical training, and started working for academia. But his passion for research never died out. He continued his research on mRNA vaccine, but it was very difficult for him to secure funding. So he tried several sources. He also applied to the California State Research Agency to secure funding for a project to develop an mRNA vaccine to combat seasonal coronavirus infection in 1996, but his application was rejected. So out of frustration, he turned his focus from mRNA vaccine to start working on DNA vaccine and on other delivery technologies. So this was not the case only for Malone. It was the case for many mRNA researchers. They were finding it very difficult to secure funding to pursue their mRNA research during this period. In fact, even companies which were interested in working on mRNA vaccines had to drop that idea and change their focus to work on other on other research ideas. Because of the conventional thinking that mRNA was very unstable, it was too prone to degradation, and also the production was highly expensive. So they did not want to invest on this. When situation was going like this, there was another land-breaking or landmark discovery which was made, and this was by two researchers, Paul Craig and Douglas Mencken, both working at the Harvard University. And what they did was they made the first synthetic mRNA using an RNA polymerase enzyme from a virus. And they went on to show that this in vitro synthesized mRNA was biologically active by injecting it into the frog eggs and showing that it was able to make the protein. So this was an important discovery. But Harvard's technology advancement development office did not think it was so important, so they did not want to patent the process. So instead, what they did was they transferred the technology and gave all the reagents to a company called Omega, which still exists, for a modest royalty, and they ironically say in a case of champagne. Okay, so when interest in mRNA vaccines was going very low and from the infectious disease point of view, there seemed to be continuing interest in the oncology circle. So here is one cancer immunologist called Eric Silvova, working at the Duke University. He had a brilliant idea. He took immune cells from blood and he made them take up synthetic mRNA which coded for tumor protein and injected them back into the host, with the idea that these cells would go back and the protein and make the proteins, which will trigger the immune system to activate killer cells to attack and destroy the lovely tumors. So Silvova and his colleagues actually they went on to demonstrate this in mice, and this approach started looking very promising. Many people got excited about this. Several clinical trials were launched. But during the course of time, there was not much success. One large clinical trial, which held a lot of hope, did not succeed, so it failed. And then even in this circle, in the enthusiasm about mRNA vaccines was coming low. However, the Silvova's work did have important consequences. It had inspired two very important people who actually became founders of two big pharma companies called CureVac and BioNTech, both of whom went on to develop COVID mRNA vaccines. So it was at this time that this Hungarian researcher, Kathleen Carrico, comes into the picture. So she came into the University of Pennsylvania. She was also passionate about mRNA research, and her goal was to transform mRNA into a drug platform. So she set up her own lab there and started working on mRNA. She started with improvising on Malone's protocol and taking it up from there. She wrote a number of grants, submitted grant application, but she had to face repeated failures. So after say 4 to 5 years, the University of Pennsylvania suddenly sent her a notice saying that it will not be able to support her entire lab without external funding. So she was given a choice of either leaving the university or accepting a demotion as well as a pay cut. It was a very difficult choice to make, but Carrico was very clear in her mind. Her passion was only research, so she accepted to stay, accepting all the conditions, but pursuing her research in mRNA. It so happened that in the year 1997, there was another new recruit to the University of Pennsylvania, and his name was Dr. Drew Weissman. So he happened to just have moved from Dr. Anthony Fauci's lab to working as a postdoc there in HIV. So these two researchers, Carrico and Weissman, happened to meet at the university's photocopying machine. They got briefly introduced to each other there, picked up a conversation, subsequently exchanged research ideas, and then they decided to collaborate. So both of them decided to develop an mRNA-based vaccine for HIV. And they did develop such a vaccine, a GAG HIV-1, GAG-based mRNA vaccine was developed, and then they wanted to test this vaccine. So Calico did an animal experiment where she injected the mRNA into mice. But unfortunately, they found that it set up a massive inflammatory reaction within the host. So the two of them were quite perplexed, but they wanted to understand the reason for this inflammation and see if they could come up with ways of overcoming this challenge. So they were looking up at the literature, trying to understand what actually triggered the inflammatory response within the host. And then they came across one paper which was published by Helminth et al. In 2000, where they had provided evidence to show that unmethylated CpG motif in the DNA, which is commonly seen in bacteria and bacteria and viruses, was able to trigger the production of type 1 interferons. So immediately they applied this concept to RNA and thought this is probably the same mechanism that is happening here as well. So they started comparing the RNA of viruses, bacteria, as well as eukaryotes, and the in vitro transcribed RNA of the synthetic RNA, which they were using for all their experiments. And they found that eukaryotic RNA, unlike the other RNAs, had a number of post-transcriptional modifications, including the presence of certain modified nucleosides, which is not there in the prokaryotic RNA as well as the in vitro transcribed RNA. So they thought, okay, this must be the key, because these are characteristic features of lower organisms, that is pathogens. The human body recognizes these as pathogen-associated molecular patterns, which are and the receptors which actually identify them and bind to these regions are called pathogen recognition receptors, or the PRRs, and there are a number of those receptors present both on the cell surface as well as within the cell, in the cytoplasm, as well as in the endosomes. So those are all listed on the slide there on the right panel. So these receptors, when they come across these unmodified RNA molecules, they are recognized as pathogenic components, and an immune response gets triggered. So there is a signaling cascade which is triggered that induces the nuclear translocation of several transcription factors, which go on to drive the expression of the inflammatory cytokines as well as antiviral cytokines, which set up the inflammatory reaction when the mRNA vaccine is developed. So, having come up with this hypothesis, they further hypothesized that by modifying these mRNAs to suit the mRNA which is actually there in the eukaryotic system, will avoid the immune system from recognizing this mRNA as a foe and triggering the inflammatory immune response. And that is what they actually went on to do. So they did a very elegant study. Actually, you should go back and read this paper immediately, and published in August 2005 by Calico et al. And here, what they did was they identified all the naturally occurring modified nucleosides in the eukaryotic RNA, and they found these options there. So they made RNA molecules containing each of these modified nucleotides. So these nucleotides include pseudouridine, 5-methylguanine, dithiouridine, 6-methyl adenosine, 5-methylcytidine. These were the naturally occurring methylated nucleosides which they identified in eukaryotic RNA. They made RNA molecules containing these modified residues and tested them in vitro alongside unmodified RNA. So that is the results of that experiment you can see on the right. So this shows the comparison between unmodified as well as modified mRNAs containing the different kinds of methylated nucleosides there. As you can see, the unmodified mRNA was producing more inflammatory cytokines, such as the interferon alpha, interferon beta, IL-12, as well as TNF alpha. This was seen in both monocyte-derived dendritic cells as well as primary dendritic cells. Next slide. We also went on to show that besides the production of these cytokines, the levels of activation that was triggered by these different RNA molecules was also significant. So that is seen there. So you can see in the first panel, the unmodified mRNA producing stimulation or activation of the dendritic cells, and how it comes down with the modification of the residues. And in a subsequent publication, which came out in 2008, they went on to show that the best response was given by RNA which contains pseudouridine. So for all the further experiments, they start to pseudouridine in the mRNA because this gave increased transcriptional capacity as well as biological stability, besides reducing inflammation as compared to the unmodified part. Next slide. So this was a very key finding, but it took time for the world to recognize that this was an important finding. However, the University of Pennsylvania was smart this time. They filed for a patent in 2006. However, they lost the smartness. In the very next year, they seem to have sold that patent rights to a very small company called Cellscript. Not knowing that this company would go on to make thousands of millions of dollars by sublicensing this patent to companies like Moderna and Pfizer in the days to come. So the reason why they did this quick change of hands was because Carico and Weissman themselves started a small startup company in the year 2007. They called it RNARX, and they tried to negotiate a licensing agreement with the University of Pennsylvania, which somehow did not work out. And that's the reason why the University of Pennsylvania quickly sold the patent rights to another company. But a few researchers actually woke up with this discovery. They thought there was promise here and started working on it. And one such person is Dr. Derek Rossi, who is shown on that in the picture there. So in 2010, he did an experiment where he showed that modified mRNA could be used to transform skin cells first into embryonic-like stem cells and then into contracting muscle cells. So this finding actually made a splash. So everybody, this news was exciting, and in fact, Time magazine featured him as one of they have this people of the year, so people who matter. So he was one among 2010 people who matter, and his experiment made a splash among the research community. And it so happened that Rossi is none other than the co-founder of the company called Moderna, the pharma company. So he, not knowing that the patent rights had moved hands from the University of Pennsylvania, went back to Ukraine, asking them to give licensing agreement with him. But only then he realized that it had moved hands, and so he had to go to Cellscript for obtaining the patent. So on the one hand, we were looking at all the developments that happened with regard to mRNA vaccines. So on the one hand, developments with regard to mRNA, and now we also have another important counterpart, which is the lipid nanoparticle, which is an important carrier for shuttling the mRNA inside the cell. So here again, there was a breakthrough in the type of lipid nanoparticle that was formed by Peter Helminth. So he working at the University of British Columbia developed a special kind of nanoparticle, which between the key to success of the mRNA vaccine, mRNA COVID vaccine. So what was unique about this lipid nanoparticle was that it contained 4 types of fatty molecules, and one of them was an ionizable cationic liquid. And why this is important is because it is positively charged under lab condition. So it could efficiently bind to the negatively charged RNA. Also, it could efficiently cross the cell membrane, which is also again negatively charged. But what happens is, and under physiological condition, it loses its negative charge and becomes neutral. So that once inside the system, it becomes non-toxic to the body. So that is a very key key point for the success of this LNP molecule. There are 3 additional LNP molecules, fatty molecules also in LNP, and these molecules are more so for the maintaining the structure and the stability of the LNP. For the first time, a person for Andrew Gill was working at Cambridge to make a combination of the modified mRNA and the LNP. But soon everybody started doing this, companies started taking it up because of the ease of production and also because the process was not very expensive. So it could be made quite simply as compared to the other vaccine. So it just can include manufacturing process just has 4 or 5 steps. So the first step is the in vitro transcription process, where you have a template coding for the protein of interest, which belongs to the pathogen, and of course you have the reaction mixture containing all the entities along with the 1-methyl pseudouracil, which is the modified nucleoside that you want to introduce, and also importantly the synthetic cap which will protect the mRNA which is going to be formed and all the capping ends. So once this reaction, the IVT reaction is over and the product is formed, the next step is purification, which is done using chromatographic techniques, and this is done to remove all the impurities as well as the double-stranded RNA which is produced during the in vitro transcription process. And also there is a small step of removing the template DNA by using DNAs for that is the template. And after the purification is complete, then both the mRNA as well as the fat molecules are both solubilized and uniformly put together in the form of nanoparticles using a technology called Mycobacterium technology with a T connector. And after the molecules, the LNPs are formed, there is further purification to buffer exchange and sterile filtering, and then there is the packaging of the sterile product ready for use. So this is the long story of the development of mRNA vaccine. So we have seen that the path to the development of vaccine was not simple and straightforward. It actually drew on the work of thousands of researchers, more than 30 years of trial. Next. So having seen about the development of the vaccine, for the next few minutes, let's look at how these vaccines act, what are their advantages, what are their shortcomings, how far we have come the use of testing or use of these vaccines and the way forward. So coming to the mechanism of action, so we know that now we have the mRNA particles which are encapsulated within the LNPs, which is the LNP vehicle. This is taken up by antigen-presenting cells wanting to be introduced into the system. So it enters the endosomes, and within the endosome, the LNP is integrated and the mRNA is released. The mRNA leaves the exosome by the process of mRNA escape, endosomal escape, and then it is identified by the ribosome and translated into the protein or antigen. This antigen can either leave the cell can secrete it out, or it can be processed within the cell. The cell's endosomal protease can process this protein into antigenic peptides, and these antigenic peptides can be presented to other immune cells via the MHC pathways, either class 1 or class 2 MHC pathways, through which they can trigger the CD8 T cell response or the CD4 T cell response to result in a robust protective immunity within the system. So what happens to the mRNA after the protein is formed is it remains for a couple of days within the system, but then through cellular processing, it is fully degraded, there are no remnants of the mRNA vaccine which will remain. It's only the memory which will remain.
Next slide.
So looking at the types of mRNA vaccines, there are two types of mRNA vaccines. One of them, one type is called a non-replicating RNA vaccine, which is a linear molecule. It has the sequence coding for the protein of interest. And as I had mentioned earlier, all the components which are essential for a stable mRNA molecule, including the untranslated regions, flagging the 3' and the 5' end, as well as the metal cap present at the 5' end and the polyethylene chain at the 3' end. So all these are important and have to be optimally provided to constitute a functional and stable mRNA molecule which can be used as a vaccine. So in addition to the use of all these regulatory molecules which we talked about or sequences, further optimization of the mRNA vaccine can happen by optimizing the sequence of the codon, which actually codes for the protein. And to increase the efficiency of translation, there is also a possibility to put in the translational mutation factor to make for better expression of the protein within the. The second type of mRNA vaccine is called a self-amplifying mRNA vaccine. So here we have an alpha virus, which is a positive strand, positive sense sequence stranded RNA vaccine, into which you can introduce your gene of interest and make the virus's replication machinery make more copies of the protein, so that with small volumes of or small doses of the vaccine, you will be able to generate a lot of protein so as to result in a sustained and longer immune response within the system. So that's the second type of vaccine. So this slide shows some of the vaccines which were developed for COVID-19. So it is not only the mRNA vaccine, there were other vaccine platforms which were also used for vaccinating individuals against the coronavirus. And looking at the mRNA vaccines, we know that the two popular mRNA vaccines were those developed by the companies called Moderna and the other one by Pfizer along with BioNTech. So these were the two popular vaccines, but there was also another company called CureVac which started making an mRNA vaccine for the coronavirus. And this company had a different school of thought. They thought that it would be safer to use unmodified nucleosides instead of using completely modified nucleosides. So they went ahead and made their vaccine containing unmodified nucleotides, but they used the same technology, the same LNP molecules for packaging and delivery. And both the vaccines went into clinical trial, but the results of the trial showed that only the perfect level of protection accorded by the CureVac vaccine was only 48% against symptomatic disease, whereas that of the Moderna and Pfizer vaccines were more than 90%. So this obviously shows that the key winner here is the modified mRNA. That's the reason why the Nobel Prize went to the inventors of the modified nucleotide. Next. So, why is mRNA technology become so popular? Obviously, because it's got a number of benefits. So, looking at the first one, which is safety, it is highly safe because there is no infectious organism which is being given delivered into the individual. It is only a piece of RNA which is not infectious, so it is absolutely safe. Can be given to individuals with weak immune systems as well. Secondly, this is not unlike DNA vaccine, which has got the risk of integrating into the post-genetic material, causing insertion and metagenesis. The risk for mRNA does not exist because it does not integrate with the post-genetic material. And thirdly, it is not produced in a bacterial system. So there is not much of bacterial contamination and reaction because of contamination in the vaccine product which is being formed. So that is yet another advantage for the creation of safe vaccine. Secondly, the time factor. The rapidity with which the mRNA vaccine was formed during the COVID epidemic tells us how fast you can make the mRNA vaccine. So it seems the first genome sequence of SARS-CoV-2 was published by China on January 11, 2020. And it appeared in 2 days, Moderna had already designed the mRNA vaccine. And in 26 days, they had the first batch of vaccines prepared. And in 65 days, the vaccine trial actually commenced. And within before the completion of 1 year, they completed vaccine trial, obtained emergency use approval, and started vaccinating individuals. So that's the speed at which mRNA vaccine can be made. Unlike conventional vaccines, which can take several years to form. The next thing is the cost factor. So the ingredients are actually very simple. So we saw that it only needed a template DNA, some nucleosides there, and an enzyme to make the RNA. Unlike making proteins in vitro, which is going to be very expensive, because several things need to be taken care of when we produce proteins in vitro. So the cost factor is also very low, which is again a huge advantage. And the next thing is the flexibility and adaptability of this vaccine template. So when you have a template or a module there, it is very easy to adapt it for any pathogen. So you can quickly prepare a vaccine in a short time whenever there is an outbreak or a pandemic. So more than everything else, all other advantages, what outweighs is the efficacy. So here we have now sufficient evidence to show that mRNA vaccines are really efficacious. So they are able to elicit the production of neutralizing antibodies which are protective in nature. They are able to trigger a robust adaptive immune response through antigen presentation via the MHC class 1 group. They are also able to trigger the innate arm of the immune response by binding to the pathogen recognition receptor. So we are overall able to prime the immune system to be able to handle an infection when it happens. So the other thing is that the efficiency of the currently existing vaccines can be made better, and there are several research activities going on to make various modifications in the existing vaccine, both on the mRNA itself as well as on the lipid nanoparticles to make better and more efficient vaccine. And another very good thing is that this is not inside any vector. So the vector includes immunity within the host, which is again a problem. Does not exist in the case of in vitro transcribed mRNA vaccines. So this vaccine has got a number of advantages, but it also does have some shortcomings. So the first thing is instability. So we've been saying that for quite some time that mRNA is not very stable molecule. However, there are ways of making it very stable by doing all these kinds of modifications, by adding the cap, by adding the pre-prime poly-A clay, by introducing nucleosides, by doing codon optimizations. These are all ways of increasing protein expression, making the RNA quite stable. But in spite of it, we have not come to the point where we can say that we have achieved a point where we can store these vaccines safely for long periods of time. As such, these vaccines can only be stored at sub-zero temperature as of now, and that too not indefinitely. They can be stored only for a few months. So the reason is because of the LNPs that are used in combination with the RNAs. So what happens is LNPs, when left in aqueous solution, they tend to form aggregates, they are oxidized or hydrolyzed, and they lose their activity. So because of all these effects, the mRNA vaccines that are currently formed are not stable. That needs to be worked on. That is a shortcoming. The other concept of immune modulation. So we will see that mRNA vaccines are capable of eliciting an inflammatory response, which is not actually wanted response. But making all the modifications like characterizing modifications of using a modified nucleoside, it was possible to reduce the inflammation to a large extent. So the vaccines that currently are in use are pretty safe. So they do not unnecessarily trigger a lot of inflammatory response. But we cannot say that they are absolutely safe, because all of you may have heard of some stray reports where people develop some complications after receiving the COVID vaccination. This is actually a true phenomenon. It does happen in a very small proportion of individuals, which also requires attention. So that is one of the challenging areas which needs more focus. And the next one is efficiency. So the efficiency of the mRNA vaccines, although good, is not up to the mark as a traditional vaccine. So they seem to elicit a much stronger response compared to mRNA vaccine, which is the reason why you need to give a number of booster doses of mRNA vaccine. So this again is an area where more research can go into. So I mentioned in the previous slide that there are some reactions or inflammatory reactions which are caused by the mRNA-based vaccine. So this is all data which is being collected from populations where mRNA-based vaccines were administered. So they found that most of the reactions to the vaccines actually happened within the first 2 days of vaccination, and it was not longer than that. And many of them, more than half of them, were only local immune responses, which were considered to be in the very mild category, such as inflammation and hyperinflammation. But there were also some systemic toxicities which were reported, and some of these include kind of allergic reactions, stimulation of autoreactive antibodies, edema of the faceless and tongue, and in very rare cases, anaphylaxis and death also have been reported. Another important observation was pathological thrombus formation, but the contribution of COVID vaccines to this is still debatable because it happens not within the first few days of vaccination. So there is no clear statistics as to the contribution of mRNA vaccines to thrombus formation. And another area of concern is antibody dependent vaccines. When we give more doses of the mRNA vaccine. And as I had mentioned, there is also one school of thought which say that the use of unnatural nucleosides is not going to be very safe, and only long-term observational studies will tell us how safe it is going to be. But that is only one school of thought. The other school of thought believes that it is safe because all the modified nucleosides that have been used are all naturally occurring modifications which are seen in the so they attributed more to the toxic recipient and the solvents which are used for preparation of the LNPs. So what is currently happening with regard to mRNA vaccine? We are progressing quite well. There is a lot of enthusiasm in the field of mRNA vaccine. The second generation of vaccines are being developed with certain innovation, and these include the use of better carrier or delivery vehicles. These are known as polymer nanocarrier, and these are believed to be more potent, have a better safety profile, and also can do better delivery of mRNA cells. So examples of these include the protein mRNA complex, which utilizes electrostatic forces of attraction for assembly, and it is believed to have superior stability and control release, and also higher loading capacity of the RNA. The other delivery vehicle that is also being researched on, which is quite promising, is a scaffold-based delivery system, which is made up of chitose and alginate gel scaffold. And another modification is the polyethylene, which is a low molecular weight molecule, which can be used for intranasal preparations of vaccines because it is a low molecular weight carrier. The other innovation is the preparation of thermostable mRNAs or lyophilized mRNAs. So I mentioned to you some challenges with the current vaccine preparation, that they are not very stable, that they cannot be stored at room temperature, as a result of which it is not being becoming available to all regions of the world. It is limited to only parts of the world where we have facilities for undertaking storage of large volumes of vaccines at sub-zero conditions. So we have to move on to animate better preparations of these vaccine candidates, or at least make lyophilized preparations which can be stored safer for longer, safer and longer at room temperature. But it's important to include cryopreservant, cryoprotectant while preparing lyophilized vaccine, and choose the best cryoprotectant. And the other areas to improve vaccine efficacy, because the third challenge we saw was the efficacy was not comparable to the conventional vaccines. We improve on that. The use of adjuvants, which is well known for a long time, also is being explored along with mRNA vaccines through administration of adjuvants like alpha, melittin, sampling in MS15 and really tested out in clinical. So that's with regard to the carrier. Now coming to the mRNA itself, multivalent mRNA vaccines are all into the book. People have started thinking about it and preparing multivalent vaccines. So these are vaccines which can target multiple strains of a particular pathogen, like the different variants that we have of the coronavirus, or it can also be vaccines which target multiple pathogens which are related to each other. This is one type of vaccine which people are looking at. The other is modular mRNA vaccine platform, which is like ready-to-use platform where you only have to keep swapping the coding gene and ready for testing for various infectious diseases. So that is something also which people are preparing. The other area of focus is targeted delivery, particularly for cancer therapy, looking at delivery systems which can target specific tissues or cells within patients. And finally, personalized mRNA vaccines, which can be tailored to an individual's genetic composition or the kind of tumor antigens present within the individual. So there are several mRNA vaccines which are in trials. You cannot obviously see them, but I just wanted to show you how many vaccines are there in trial, how much of mRNA research is currently ongoing. You can see vaccine candidates which are being tested for several viral infections as listed there. You can see that there are vaccines for HIV, for respiratory syncytial virus, for influenza, for Nipah virus, next slide, for Zika virus, for Epstein-Barr virus, for a number of cancers. So there are many vaccine trials which are ongoing, and they are at the various stages of clinical trial. You can see them listed there at phase 1, 2, 3. But most of them are in phases 1 or phase 2. We only have 2 vaccines which are currently in phase 3. Next slide. Okay, so this is a snapshot showing all the vaccines which are currently in the pipeline for both for vaccines as well as therapeutics or the mRNA pipeline. So there are 138 candidates in total, but 104 of them are in the pre-clinical stage of testing. The remaining of them have all already entered clinical trials. And as I had mentioned, there are 2 in phase 3 trials, about which I will show you in the next slide. But you can see here that the pie chart shows that most of the vaccines which are produced are against infectious diseases, and another big chunk is for cancer. And that is the bar there shows the companies which are actually the industry which are pioneering on mRNA vaccine. So you can see Moderna at the forefront, having 29 candidates already in trial and 7 more candidates which are being prepared. Next slide. This is very interesting for me because, unlike what happened in California's time, Malone's time, where funding for mRNA research was very poor, now funding for mRNA research is really improved. So it's not only coming from industry, it is also coming from the government sector, which is actually a good thing. So investigators, researchers passionate about mRNA, this is the time for them to actually take a plunge and do some very good research. So these are the two vaccine trials which are in phase 3. The first two of them. The first one is by Moderna. It is the RNA-1010 vaccine against seasonal flu, and there is another vaccine also for seasonal flu developed by Pfizer. Both of them in phase 3. The results are expected in 2023. Moderna has given some indication that the results seem to be positive, but the final report is yet to come. Pfizer, however, have not declared into the results till now. The study is still ongoing. There are 2 other vaccine trials which will be completing this year, from end of 2023. One is a phase 2 trial for advanced melanoma, and another is a phase 1 trial for HEPA. And both these trials also seem to be promising. So the future. So now we have seen that already vaccines are being tested extensively for infectious diseases as well as for cancer. But there is more to mRNA vaccines. They can be exploited for treating allergies by inducing tolerance to specific allergens. We can also use them to prevent or treat autoimmune diseases as well as metabolic conditions like multiple sclerosis, rheumatoid arthritis, type 1 diabetes. There is also scope for using mRNA as a drug for treating various genetic disorders like sickle cell anemia, hemophilia, cystic fibrosis, where a deficient protein can be administered to the body. And there is also hope that mRNA will find a place in regenerative medicine, allowing programming cells to produce specific growth factors, a signaling protein, which can be used for repairing tissues, like for conditions like heart disease, spinal cord injuries, as well as regenerative disorders. So the scope is very broad, and horizon is really large before us. So that's where we are at the moment. But from all this, I just want the students here to take this message home. So what we learned from all this story about the mRNA vaccine is that it takes decades of hard work and toil, disappointment at times, dead ends, rejection, battles, over profits, over patents, but then one needs to continue and prevail and succeed with the generosity, curiosity, and persistence against all kinds of skepticism and doubts. Faith in your passion, faith in your dream, and the passion to pursue it will definitely bear fruit. So a portrait about whom we saw, the person who actually made the first synthetic mRNA, he says that it is a new discovery for that matter. It's a long concept, and you never know what's going to be useful. So just keep doing. You have a goal, keep pursuing it. Something useful will come. At no point, you shouldn't keep getting frustrated, like how Melon did next. So here is a very humble message by the Nobel laureate Calico. After being announced as the winner, she says, everyone just incrementally added something, including me. So sweet of her, but we know from the story how much she went through and how she persevered and how she got this. So all of us salute her and take home a message from her of hard work and perseverance. Thank you so much for taking us.
PSL and other things.
That's an excellent lecture. So let us go for the question and answer session.
No, the PSL and other things at minus 70 degrees centigrade, right? Yes. To call it sub-zero is bit true, but 70. See, that is a serious problem. And, it cannot be done other than Western countries, US and other places. And what about the Russian one?
Minus 70 for 6 months.
Ah.
Minus 20 for 3 months. So ideal is minus 70.
Yeah. And what about, there was something in Russia also. So we
Yeah.
What?
So there was the Sputnik vaccine is from
Fine.
Russia. So I did show it there. The Sputnik vaccine is not an mRNA vaccine. It was viral vector based vaccine. That's a Russian vaccine. The Covishield vaccine that was available in India is also a viral vector based vaccine. The Covaxin that we had, that is an attenuated viral vaccine.
Any questions?
Other than the US, what are the other countries who are doing this type of mRNA? In India, do we do?
No.
So actually, no. If you look at the 138 candidate vaccines which are there in preclinical and clinical trials, none are from India. The vast majority are from the US, some from Europe, there are some from China.
China also doing?
Yes, China has a number.
No. I understand that so far we have not done it yet, but at least now is there any program of doing like that? Not yet.
So we are not aware because there have not been publications yet, and none of them have come to the stage where they are ready for clinical testing. So we are not aware, but probably there is.
We have some technology and knowledge.
Of course, sir. The production is like not at all a complicated process. It doesn't require great technology to do it. So it's definitely possible.
So among the challenges that you have noticed, there were immune modulation, one of them. So some of them has, you also said it gives a lot of immune response as suppress aggressive reactions we have noticed. Though also that you have noted that it must be given attention. A few proportion of people do you have any idea as to like how do we overcome that? Like how can we modify or invent further vaccines in order to overcome that problem as well?
So actually, as I mentioned, people have come to sort of believe that these inflammatory reactions are not triggered by the mRNA itself. It possibly is because of the solvents and excipients which are there during the preparation of the complex, the LNPs as well as the mRNA complex. So that is the one which needs modification. And additionally, it could be the LNP itself. So that's the reason why people are looking at better carriers with a better safety profile, which can be used in the subsequent vaccine. Because as you rightly said, even if it is a very small proportion, it's not something that can be ignored. We also need to keep working towards perfection, but there is research going on.
Excellent talk. Some people are getting what they call long COVID, some 5% or 10%. So are there any suggestions for such patients? You know, they are getting dizziness and inflammation due to exertion, post exertion syndrome. Since long COVID is people are studying that. But, Do you know any, can they take another vaccine or like?
Yeah, this problem is there. More frequently than what we actually realize. Because when you talk to people, a lot of people say they have, lasting effects of COVID. But then there are actually, as far as I know, there are no treatment modalities as such for treating long COVID. It is just management for the condition at that point of time. And when it comes to vaccines, actually they are known to protect from severe disease. But once the infection is already there, the additional protective value of the vaccine itself is quite debatable. So how much more protection will a vaccine give for a person who's already had 2 exposures? So you don't have an answer to that. People who are already infected and they have these symptoms which keep troubling them. I'm sorry, I did not have an answer.
I have one question on regard to the booster vaccines. Why do we need to go for boosting? So, successive boosters are required. And another question is now, if you are using your mRNA vaccine, you can drop it and go for another conventional vaccine during your booster vaccine.
So to answer your first question about the need for giving booster doses. The mRNA, as I said, it's got a very short half-life within the body. So it just stays there for a couple of days and then it is secreted by the post-cellular processes. It's gone. Within that time, it makes a protein and then it generates a immune response required to get protective immunity. But this is not, as I said, the durability is, we still do not know because it's only like 2-3 years after the mRNA vaccine came, and with whatever knowledge we have, the response does not seem to be long lasting. That's the reason why we need to have repeated boosters. So, what would overcome this is the use of a self-amplifying vaccine. So, in that case, the virus backbone that you are using for inserting the pathogens protein is actually capable of replicating, transcribing, making proteins constantly within the system. So there is a longer or sustained immune response which is generated, which is not the case with the conventional mRNA vaccine. Then your second question about
combining these two
vaccine. Yeah, you take one vaccine, is it okay to take another vaccine? So that is allowed now. Changing the types of vaccines for the subsequent boosters. They sometimes say heterologous vaccination gives even better safety profile as compared to the homologous vaccine. So that is something which people have thought about, looked at in a little bit of detail and say it is all heterologous vaccination.
I have got this. I really do not know so much about it. You said that. The mRNA that itself is unstable. But once it is there and it served its purpose of producing sufficient antigens capacity for the immune cells here. Then why do you bother about the stability of the mRNA? Because already your body has achieved the capability to do.
Okay. So when I was talking about stability, I was referring to stability prior to vaccination. So during the storage.
Till it is getting delivered to the.
Okay. So you prepare the vaccine, you need to store them, and when you deliver them, they have to be in effective form. They cannot, decompose, they cannot aggregate, they cannot lose their activity, they cannot become oxidized. These are all the challenges that I was talking about. So within the system, of course, like what you said, once the job is done, there is no need for them to stay any longer.
One question from YouTube. If no one from floor, can I do that? Yes. do we have any effects on telomeres such as shortening when we use the mRNA vaccine? Telomeres. Do we have any effects on telomeres such as shortening when we use the mRNA vaccine?
I know, I got a question, but I didn't have the answer for that. I have not seen any papers which talk about telomere shortening with regard to vaccination. So I'm sorry, I didn't know.
Further additional question to what sort of posed, are there any adversities associated with booster vaccines? Like once you said we are allowed to take multiple vaccines. Of course, they have carriers and other toxins associated. Will that not affect the individual system, like additional intake? Will that not affect the system of any individual?
So if you're talking about mRNA vaccine, there is no mRNA accumulation there. So it just is gone. In a couple of days, it's gone. However, there is one problem which people refer to as antibody enhancement. Yeah. So, constantly, repeatedly triggering the immune system can cause cytotoxicity because of buildup of antibodies. So that is one issue which is talked about, but it is not widely reported. So we have had people already taking 2, 3, or even more boosters, but antibody-dependent cytotoxicity has not been actually reported that one. Actually. Antibody-dependent enhancement, not toxicity.
I have one more question on your last slide. The last slide, you have enlisted a list of difficulties and challenges in which battle for profit, so many things are enlisted. Again, why does it happen only in the research on the medicines or mRNA? Ribosome, there is no. Of course, there is a rivalry, but it doesn't manifest in the form of grabbing profit or something like that. Right? So again, the investment is coming from the private sector, the companies. So many things are happening. How are we going to relate those things? It's a challenge to the scientific community, right?
Probably because there in every field, this just comes to the light because of the magnitude of the profit. Probably that's why it becomes the showcase. Probably just there at a smaller level, but in every field, I'm not sure.
No, other researchers are. This is also a fundamental science research on fundamentals.
Actually, they do.
But this research on fundamentals is related only a specific field have such challenge.
Also, that the graph that you showed had sponsorship from government to be at its peak only during 2021, that is post-COVID, that is affecting a large population. What is your take? Why has it reduced? I mean, after 2022, 23, I mean, it's important to,
Interesting. But 23 is not complete yet, no?
Yeah,
We have another year.
Again, it's
Have
not
another 2 months, let's hope it will cross.
again, we see the trend is industry sponsor is more, right?
That means more profit, right?
Yeah, the government, yes. That happened.
one of the major problem that we such as
So the government program we have, no, for 5 years, this much funding.
So unless it is a life and death scenario, okay, thank you, okay.
Okay, we can wind up.
Thank you
Thank
so
you.
much.
So
Thank you.
I invite Dr. Murugan to
It is really pleasure to thank the speaker. Wonderful speech, very inspiring, connecting social relevance of the whole idea of mRNA vaccine, comparing with other vaccines. I was really very much surprised to see that discussion so much on patenting and intellectual properties are very much a part of this biological science. The last 40 years and 50 years compared to the earlier, it was a physical science. For the last 40 or 50 years, it's the biological science because of the sheer, as you say, correctly, as you pointed out, sheer amount of scale of the problem. Very nice. Thank you. Excellent topic.
So that's it. I would like to thank the audience. We'd like to thank the audience for coming all the way and brought the audio programs and listened to this lecture. And the advice and at last, I would like to remind you of this. Fill up your feedback form. It's available in the link. Just click the link and fill up your feedback. And again, I invite you to become a member of Mycobacterium Science Forum and take the Thank you. Next program. Our next program is going to happen in instead of analytical sciences, it's going to be on Nobel Prize. It's going to be delivered by the Shorama from the